ये फोन स्पीकर के पास क्या है हॉटस्पॉट चला
day, one and all. I'm Kasturi Kelly Reddy, and along with Nikhil Nishavam, we would like to cordially welcome you back to the 7th International Symposium of the New Frontiers in Cardiovascular Research. We are now in the session 3, where the topic for discussion is inflammation, thrombosis, and vascular disease. This session will be from 2:30, well now 3, till 5:30. I would like to welcome our chair for this session, Professor Klaus Presner from Kirchhoff Heart Research Institute, Department of Cardiology at Justus-Liebig-University, Germany. Professor Presner studied biochemistry in Cologne, Germany, and there he received his PhD in biochemistry in 1978. He was a postdoctoral fellow at the Department of Immunology at the Scripps Clinic and Research Foundation from 1980 to 1983 to work on regulatory pathways of the complement system. Thereafter, he became principal group leader at the Clinic Research Unit for Blood Coagulation and Thrombosis of the Max Planck Society in Giessen from 1983 to 1997, followed by the position as associate professor in 1987 at JLU in Giessen. From 1998 to 2009, he was professor for biochemistry and pathobiochemistry and head of the Department of Biochemistry at JLU Medical School in Giessen. Thereafter, and until now, he serves as senior scientist at the Kirchhoff Heart Research Institute, Department of Cardiology, JLU Giessen. Please can I have a round of applause for Professor Presner.
Now, swiftly moving on, we'll be presenting Professor Sean Jackson. Professor Sean Jackson is an NHMRC Senior Principal Research Fellow, a faculty member at the Scripps Research Institute in Las Vegas, San Diego, and an honorary visitor fellow at the University of Cambridge. In 2015, he was appointed as the inaugural Director of the Cardiovascular Research at the Heart Research Institute and Charles Perkins Centre, University of Sydney. He has a clinical appointment in the Department of Cardiology in the Royal Prince Alfred Hospital. He has established several Australian biotechnological companies focused on the clinical development of novel antithrombotic therapeutics. Professor Sean has received numerous international awards, including the Marison Bernhardt Prize in 2009 and a Career Investigating Award, and alongside with a medal in 2011 from the International Society of Thrombosis and Hemostasis. So we would like to welcome Professor Sean Jackson on stage. He will be presenting on a red cell endovascular seedants causing COVID-19 microangiopathies.
Right. Thank you very much. And let me begin by thanking the organisers for the opportunity to come. This is my first time to Mauritius, and I tell you, this has been an amazing experience. The people are just so welcoming. And when I came in today, saw all the volunteers, all the young doctors of the future, I was so impressed with the enthusiasm and excitement about having this international symposium here at the college. Klaus Presner and I had dinner together in 2019 in Melbourne, Australia. I'm originally from Melbourne, and Klaus told me about the Frontiers meeting and the places around the world where you have the meeting. I was quite excited. But of course, the pandemic got in the way, and I've had to wait 3 or 4 years to be here. So thank you to the organisers for the invitation. Now, my background is very much thrombosis, play the research. So I'm going to give you a talk today that I'm not used to giving. And I have to say, it's one of the most interesting and unusual studies we've ever done in the lab, and it was completely unexpected. I'll go to the first slide. Oh, I can do that. Now, this slide, you all know the Sydney Opera House. This is to remind me that not only that I live in Sydney, but it's a very special week in Sydney because this is the 50th year this week of the official opening. And many of you know this was an amazing feat of architecture and engineering. It was a very famous Danish architect by the name of Jørgen Utzon who came up with this incredible design. And it was supposed to be built in the 1950s, was to be built over 4 years and to cost $7 million. The reality is it took more than 20 years to build and cost well over $100 million. And back in the 1950s, that was a lot of money. So the lesson of the Sydney Opera House is one has to be very patient, and you can produce things that are quite remarkable. And I'm sure many of you have seen the Opera House. If you've had the pleasure of visiting the building, it's quite a remarkable structure. Okay, so COVID, this audience, I'm sure I don't have to give much background. Obviously, it's a respiratory illness, but what causes many of the deaths are systemic effects. And essentially all organs from your nose to your toes are affected by COVID. We know now that the systemic nature is related to a microangiopathy, and that this microangiopathy can lead to multi-organ dysfunction. And the endothelium is really the key part of the mechanism of this microangiopathy. And the term endotheliitis is used as a fairly nondescript term. Early on, it was thought that this was probably due to the direct effects of the virus through the ACE2 receptor on the endothelium. I think there's growing evidence now that it's probably multifactorial. The cytokines play an important role in damaging the endothelium. Complement. There's very good evidence that complement's playing an important role in microvascular injury. And increasingly, evidence that hypoxia and ischemia plays an important role. And that's very important in what I'm going to talk about today. So endothelial dysfunction and microvascular obstruction, obviously the endothelium in its healthy quiescent state has very important anti-thrombotic and anti-inflammatory properties. And with endothelial injury, the endothelium becomes pro-thrombotic and pro-inflammatory. And complement, coagulation cascades, platelets, neutrophils are thought to be key mechanisms for microvascular obstruction, as shown in this schematic diagram. And COVID coagulopathy, and as a hematologist, it is considered one of the most aggressive coagulopathy, causing both small vessel microthrombi as well as large vessel arterial and venous thrombi. Now, this struck me when I looked at this study back in 2021. It's called the MYSTIC study, where patients who are in intensive care, either ventilated or not ventilated, the microcirculation under the tongue was visualised using intravital microscopy. I love intravital microscopy. And what was striking to me was the degree of microvascular obstruction. So in the smallest capillaries, below 10 micron in diameter, up to 90% of those capillaries was obstructed. And even when you follow these patients that recover out 18 months and develop long COVID, they still have a considerable degree of microvascular obstruction. And the theory is that this is probably due to excessive coagulation in the microvasculature, fibrin buildup, and this leads to long-term blockade of the microcirculation. However, despite overwhelming evidence for a prothrombotic state with coagulopathy, anticoagulant antiplatelet therapies have had limited impact on the disease. And this has raised the question, are there other mechanisms? And this is where we came into this story. We wanted to re-examine the endotheliopathy and correlate that with the microvascular obstruction in major organs from COVID patients. To do this, we obtained 8 COVID patient postmortem specimens. This is from patients who had varying degrees of multi-organ dysfunction, and we examined the major organs, the lungs, kidney, liver, heart, gut, etc. Unfortunately, we didn't have brain samples from these patients. And Amy Wapkowitz and her colleagues had done the detailed autopsy and histopathology findings and demonstrated, as in many other studies, that there was a lot of platelet and fibrin thrombi in these organs. So the first question we want to look at was the nature of the endotheliopathy and look at it in some detail. And I've got to give credit, there were 4 people in my lab that spent 2 years looking at this. So they've gone to great lengths, and it has been easy to do. We started off with the standard endothelial surface markers, molecules like PCAM, cadherin, glycocalyx, and we confirmed what others had showed, that you lose a lot of these in the microcirculation. What we didn't expect, though, was the nuclear changes. And on H&E, if you look carefully, you can see a dying endothelium. There are typical nuclear changes, such as pycnosis, karyohexis, and I'll explain that a little bit more in a moment, karyolysis. So what we found is many of the microvascular where you had endothelial death, you had red cell abnormalities. It was a very close correlation. And when you look carefully at the endothelium of these vessels, you would see the nucleus becomes irregular and condensed. That's classical epignosis. You would also see fragmentation of the nucleus and complete lysis. And what was striking when you looked at the lung, liver, kidney, and heart, and I should emphasise this, we didn't look at areas of infarction deliberately. We didn't look at areas where there could have been autolytic change, post-mortem change. We looked at viable regions of these organs where there was clear damage. And what we found is that in up to 60% of vessels, there was dying endothelial cells based on those nuclear changes and loss of those circular markers. That was quite an unexpected finding, that there'd be that much endothelial death in the microcirculation. Not only were these cells dying, you can see that there is endothelial cell detachment. So the H&E on the left, I can use the, what have I done here? There's a laser. That's this one here. Okay, so you can see here there's an endothelial cell, there's another endothelial cell. This has PCAM, it's an endothelial cell, but you can see it's detached from the surface of the vessel. And we saw that in at least 20% of vessels. That's a lot. That's a lot of endothelial attachment. The other thing that was striking, where there was endothelial death, we could see this amorphous material. There were damaged red cells and this amorphous material. When we asked our pathology colleagues to look at this, they said, yeah, we see that quite often. We call it amorphous acellular proteinaceous material. We said, well, what is it? And they said, we don't know. It could be fibrin or it could be other proteins that precipitate in blood. So we were very keen to understand this because there was a very close association between the endothelial death and this amorphous material. Under EM, it was unclear what this was, so we went and did multiplex immunofluorescence. And this technically is quite challenging, where you stain the same cell, the same tissue with antibodies against fibrin, platelets, nuclear stain, because we thought they may be nets from neutrophils, and we then looked at red cells. And to our great surprise, this material was all derived from red cells. And there was one feature that when you have hemolyzed red cell membrane, the antibodies we used against proteins such as glycophorin and BAN3, the fluorescence lights up. So you get a much stronger fluorescent signal. And I'll show you again on the next slide. So there was a lot of this material in the COVID microcirculation. When we looked at regions with intact red cells, you can see that here, this is the H&E, there's this amorphous material between the red cells. And with the multiplex immunofluorescence, you can see that this is lighting up, it's much stronger staining, whereas the intact red cells, you see it's positive, but much lower intensity staining. So this was a very helpful way to pick these hemolyzed red cell membranes. And you can see that it's acting as glue between the intact red cells. This is also. Now, the other thing that surprised took a long time to be convinced that this is reality is when we looked at the amount of platelets, fibrin, and hemolyzed red cells in microcirculation. We looked at the liver, kidney, lung, gut, heart. And what we found, this is an example in the liver, there's widespread hemolysis in the liver. There are some platelet microthrombi, but quantitatively much less than hemolyzed red cell, and not a lot of fibrin. Most of the fibrin was in larger vessels, greater than 50 micron. The smallest vessels were chock full of these hemolyzed red cells. So the conclusion from these studies is there is extensive endothelial death in the microvascular of COVID patients, in the kidneys, liver, heart, gut, lungs. And at the sites of endothelial death, prominent red cell hemolysis. And these hemolyzed red cell membranes promote red cell aggregation and microvascular obstruction. Now, I mentioned before about the importance of ischemia. What we noted was the COVID patients that had evidence of ischemic liver, kidney, such as acute tuberous necrosis, gut, or had acute coronary syndrome, they had the most extensive hemolysis. And so we then looked at patients, non-COVID patients, who had had either a stroke heart attack, gut ischemia, and we saw widespread, the same phenomena of widespread hemolysis and microcirculation associated with endothelial cell death. So we then went to experimental model to see if we could reproduce this. So we used two models. We used a stroke model in the mouse, an ischemic model of stroke, and we used a gut ischemia reperfusion model. I won't show the data from the stroke model, I'll show the gut model. And what we find, if you look at, this is a simple villus of the gut. Typically when you use differential interference contrast microscopy, the intact red cells are dark. The majority of the vessels were translucent. And when you did the immunofluorescence, you could see that this primarily reflect live red cells. And those live red cell membranes were associated also with the intact red cells. And this was associated with both the ischemic and the reperfusion phase of injury. So is microvascular hyperperfusion linked to endothelial cell death? And then the question is which pathways? And so we developed a model where we could look at endothelial death at the whole organ level. I won't explain all the details, but we use a Nexen staining, which binds to phosphatidylserine as a marker of apoptosis. And we found that there were two regions of staining. There's a high areas of high annexin binding, and these areas of low annexin binding. And when we used a series of markers of apoptosis or necrosis, the high annexin binding regions were apoptotic endothelial cells, whereas the low regions, these had features of necrotic cells. So there was a lytic form of cell death. And then when we looked at the microvascular obstruction, we want to correlate endothelial death with microvascular obstruction. We find there's 100% correlation. So where you have apoptotic cell death is where the platelets occlude. And if we do a fibrin stain, that's where fibrin is as well. Where you have necrotic endothelial death, it's exclusively mediated by hemolyzed red cell. So we're intrigued by this, that you've got two distinct forms of endothelial death inducing microvascular obstruction through distinct mechanisms. So we spent the last 3 years trying to work out how that occurs. And that's quite a fascinating story. Now, cell death is a really topical area, particularly the various pathways leading to necrotic cell death. Historically, we think of necrosis as just accidental cell death, due to hypoxia, due to radiation or chemotherapy, etc. Whereas apoptosis is a regulated form; it's a programmed cell death, genetically controlled. Well, our concepts have changed a lot. We now know there are many forms of regulated necrotic cell death. The most, famous of those is necroptosis, and that involves a series of kinases, the RIP kinases, leads to the formation of this oligomerized complex called the MLKL complex, which essentially punches holes in the membrane. That's why the membrane's live. So as I'll show in a moment, phosphorylation of this is a very good marker of necroptosis. I won't go into the other pathways. We've heard a little bit earlier today about mitochondrial permeability transition pore, but I'm going to focus on necroptosis. So we used an antibody against the phosphorylated form of MLKL, and what we found is in regions where you have red cell hemolysis and endothelial necrosis, we got a positive signal for necroptosis. So to confirm that this in fact was necroptosis, the most definitive way to do that is to study MLKL knockout mice. So we were interested in the question, if you prevent endothelial necroptosis, do you see red cell hemolysis and microvascular obstruction? So we answered the first question, that is, there is a dramatic reduction in red cell hemolysis when you stop endothelial necroptosis. And we've done conditional mice, so we've knocked out MLK in the endothelium only, and we see exactly the same thing. So endothelial necroptosis is important for hemolysis. The thing that really surprised me was I would have thought if you were having less dying endothelium, you'd have less bleeding because the endothelial cells should be preserved, that should be intact. We saw the complete opposite. And we saw this quite massive interstitial bleeding in the MLKL, which was a big surprise. So we then wanted to understand a bit more about what was triggering hemolysis. I was pleased to hear that Klaus Love is live looking at complement. And of course, complement, intravascular hemolysis, a number of diseases where that occurs. And it is well known that dying endothelium is a potent activator of the complement pathways. Then there's three pathways leading to complement. Dying endothelium can induce that. And it's well known in COVID, there's a lot of complement activation in the microcirculation. So complement is a very complicated field. I'm not an expert, but one of the ways to complement its innate immune function is through the formation of this thing called the. I didn't hear you. Okay. There's this thing called the membrane attack complex, which is what punches holes in bacteria. So we wanted to look at what happens when we block that complex, and you can do that using mice called the C9 knockout mice. And the C9 knockout mice, so that up the earlier steps in complement activation, C3, C5 is all intact, they just had trouble forming the membrane attack complex. And the phenotype from these mice is identical to the MLKL. You don't see hemolysis, but you get this massive interstitial bleeding. And again, that was a huge surprise to us. So, does necroptosis regulate microvascular bleeding? Obviously, the question based on the knockout is yes. So, what's the mechanism? Well, we look very carefully at where bleeding occurs. So, if you look at ischemia in the villi, you actually get regions, mostly obstruction, but you actually see regions of bleeding as well. And what we noted was the regions where there is hemolysis obstruction, with very little bleeding there. Regions where there was little hemolysis, we saw bleeding, and there was a reasonably good correlation between the two, suggesting that the hemolytic process is having some sort of hemostatic function, which intrigued us even more. Okay, so what's going on here, and this is why in the title I've got this concept called endovascular sealant. What's happening is that as these endothelial cells die, activate complement, the red cells start to lyse, and I'll show a video at the moment what happens to these membranes. They get stretched and form this layer on the vessel wall. And this layer builds up. You'll see it more clearly on the video. And what you see here, these are intact red cells. These are these layers of sealant that's forming and preventing bleeding. When you have a TM, for example, you can see these are intact red cells, and you see this. This is all we've done in CLEM, which is correlating fluorescence with EM. This all lights up. This is all red cell membrane here, lying on the wall. So how does it form? So here's the video I just mentioned. Hopefully I can get that to work. You know how I did it? So the first video is prior to ischemic injury. And what we've done, we've labelled the endogenous red cells, Alexa green, and we've used a doctor transfer experiment. So about 5% of the red cells we've infused have been spiked with Alexa 456. So if you go to the next one, if you don't mind. So after IR injury, what you're seeing here is these sealant layers. If you look at this red cell coming in here, you can see that this membrane is getting stretched and forming this layer. And these membranes are really thick, so they attract other red cells. So you get this progressive build-up of this sealant layer, which becomes the hemostatic layer to prevent bleeding. And of course, when this becomes exaggerated, red cell hemolysis, and this, we've got a nexus here, so this is where the endothelial death is occurring. This is where the hemolysis is occurring. We get that increased intensity of the red cell membrane. There's three stages. There's the initial sealant layer, and then the red cells that act, that have membrane associated, get sticky, forming these aggregates, and eventually you'll get total occlusion and total hemolysis in regions of vascular. So we think that this is a new form of microvascular obstruction in patients who have died from COVID, so severe COVID-19. What our studies suggest is that in contrast to the conventional thinking where you might have endothelial death, you'd have a lot of fibrin, what we see is that at sites of endothelial death, you have hemolyzed red cells. And that the Dying endothelium activates complement, which is the mediator of hemolysis, and that this, I didn't show the data, but you don't need platelets, you don't need fibrin for this hemostatic mechanism. So this is a distinct hemostatic mechanism mediated by red cell membranes, and when excessive can lead to microvascular obstruction. And this, I think, has some interesting therapeutic implications. That if you target red cell hemolysis, either by targeting necroptosis, complement, or potentially some other mediators, this potentially could be a new way to reduce microvascular obstruction. I just want to finally acknowledge the two people that drove this work. Mike Wu, who's a postdoc in my lab in Duke-NUS, who's worked with me for many years. This has been a big team effort. Many people in my lab. I mentioned Amy and Tessa from New York, who's been really helpful with pathology work, as well as our pathologists at the Alfred, and our colleagues who provided us with the various knockout mice. Thank you.
Thank you very much, Sean, for this great talk with really novel, unexpected data. So, before I open the discussion for just a few questions, something crossed my mind. We were working on angiogenesis since 30 years, in those days in the 90s, working out mechanism of diabetic retinopathy. And we saw very, very similar pictures, but couldn't believe it, it really didn't follow it, that there were partially destroyed red blood cells in these more or less avascular structures, etc., very similar to what we have seen there. So that's really interesting. What can you compare? What?
Yeah, so we've looked at diabetes in this model, the gut IR, and you get much more dramatic hemolysis. So, you know, in diabetes, red cell abnormalities are a lot more common. I suspect endothelial depth is with ischemia is more severe as well with diabetes.
So you would also agree to call them avascular or antivascular capillaries, because they do not really see blood and there's no fluid inside.
So we've infused dextran to see just how porous, and we compared that with platelet thrombi. That's what we regularly do. And platelet thrombi dextran can usually penetrate, but when you get the hemolysis, you cannot. So those vessels are completely not getting in. I suspect platelet proteins.
So please open for questions.
Thank you. Your sealant or amorphous material you refer to as lysed red blood cells. So are those membranes or membranes and hemoglobin or only hemoglobin? So what is this material? And secondly, is there a hemoglobin in the blood of those patients? Because with all those lysis, even though it may obstruct some microvasculature, you might expect to find hemoglobin in the bloodstream.
Yeah, they're really good questions. So there are some reports early on in COVID where they saw schistocytes in the peripheral blood. And as you probably know, schistocytes is part of red cell fragmentation, which we typically see, my background is hematology, we see that in DIC, disseminating coagulation. So the authors at the time, even though, as you know, DIC is unusual in COVID, it's not common. So the authors at the time suggested that this was a fragmentation of red cells because of endothelial damage. So they thought it was in keeping with a microangiopathy. You make a very good point about hemoglobin, because you would expect to see hemoglobin reduced, increased by bilirubin, for example, maybe even reductions in haptoglobin. We've done anti-hemoglobin staining, and obviously the intact red cells light up. All the lysed membrane, there's no hemoglobin left. So they're completely lysed. You do see partially lysed red cells. What happens, as you know, heme is incredibly toxic to the vasculature. So I suspect this is exacerbating the injury that's occurring. How much is circulating? It can't be at very high levels. People would have picked this up long ago. So this presumably is a very localized hemolysis. and you're not, going back to Klaus's point, you're not flushing this tissue out. It's blocked completely. So maybe, I don't know whether that's part of the explanation, but in our animal model, we can certainly pick up hemoglobin circulating, but not at very high levels. Thank you.
It's a very fascinating discoveries. And, yeah, congratulations. So I have two questions. So the first one, is the phenomenon what you observed specific to COVID, or is it just general to any virus infection? The second question is, what caused this endothelium necrotosis? And if there's a spike protein or just a general cytokine storm?
So we haven't looked at any other viral infections at this stage. As I mentioned to you, we've looked at patients who had ischemic heart disease, stroke, gut. We looked at colon, and it seems to be a consistent feature of ischemia. So I don't know that you need the virus. I think it's a reflection of ischemia and hypoxia. Obviously, endothelial cells are very sensitive to ischemic injury. And I should say, we start to see this following 15 minutes of ischemia. It's quick. You start to see death of endothelial cells and red cell deposits. So your second question was about
the mechanism. So what caused this phenomenon?
Yeah. So I've focused on necroptosis, but we have some evidence that the cyclophilin D dependent necrosis is also playing a role. And that's not necessarily surprising because there's quite a degree of redundancy. As you inhibit one death pathways, other death pathways can kick in. So necroptosis is obviously important to this phenomenon, but we have some evidence that other death pathways. And it usually relates to the degree of the insult. And there's presumably going to be a fair bit of oxidative stress occurring here as well, which is going to increase the mitochondrial permeability transition for. I don't know, there is some work on ischemia and necroptosis. There's some evidence that acidosis activates ion channels, which activates RIP kinases. But it's not a well-studied field at this point.
I mean, this virus is mostly in the lungs and, it gets into the lungs. I'm just wondering, how do they get access, to this blood vessel, like endothelial layer? Is this spike protein circulating around, or is just, the cytokine storm because of this complement activation, etc., etc.?
I mean, we haven't looked at cytokines at all. In the ischemia model, 15 minutes, I would doubt that that's long enough for this to be driven by. So we've looked at TNF knockout mice, doesn't block it, and we've looked at TNF receptor knockout. So it's not to say there are not other death receptor molecules or ligands involved, but we're not 100% convinced that you need cytokines for this method.
Thank you.
Okay, but Derek, very short one, please.
Thank you. I really enjoyed that. You mentioned necroptosis. What about from an inflammatory point of view, pyroptosis? You look at that question.
Yeah. So we haven't. Pyroptosis and ferroptosis. I think these are really interesting questions. I did mention about cyclophilin D dependent mitochondrial permeability transition fork. We've also got preliminary data with BAC and BAC's knockout mice. So if you block apoptosis, maybe it's surprising, you get a massive bleeding problem as well. So there's some link between endothelial death. I mean, I don't fully understand this. I mean, inhibiting death should protect the vasculature, but for some reason, we're triggering this bleeding problem. So lots more work to be done.
Just to finish up the discussion with a very short question from my side. Have you had the opportunity to analyze the fibrin network, the fibrous material in those vessels?
So we've done scanning electron microscopy at a very high level. You can, and correlated that with the fluorescence, so we know exactly what's fibrin. It is different from the red cell membrane, but to an uneducated eye, you wouldn't necessarily pick it visually. And that's what pathologists said. Normally they consider that fibrin, this stuff, but it is different.
Well, we found last year, was published that the fibrin material with severe COVID-19 picture is much denser than the regular one, first of all, due to increased factor 12 activation complexes. And secondly, this fibrin material is much more resistant against thrombosis. And this together could be the reason why there is a higher risk of thrombosis.
Good point. Thanks, Ken.
So, thank you very much. And
Yes, thank you, Professor Sean Jackson. Now I'd like to call upon Elizabeth Nye, who will be talking to us about the impact of the cold-inducible RNA binding protein, CIRP, on arteriogenesis.
Good afternoon. Good afternoon, ladies and gentlemen. I'm very happy to be here, and of course, I want to thank the organizing committee, Klaus, Jerry, all the lovely organizers for inviting me to come here to Mauritius and to present my data on Arteriogenesis in front of this audience. So the title of my talk is Impact of the Cold-Inducible RNA Binding Protein SIRP on Arteriogenesis. So, coming first to the background and goal, of course, we have heard today several times that vascular disease are still the major cause of mortality and morbidity worldwide. So current treatment options are, as we also have heard, balloon dilatation, stent implantation, and if this doesn't work anymore, if it's not possible anymore, we have a bypass surgery. And Gary said this morning, now it's time to get new therapeutic options to help those patients. And the idea is, and the goal is to develop non-invasive options to treat those patients with cardiovascular occlusive diseases. And the idea is finally to promote the growth of a natural bypass which compensates for the loss of function of the occluded artery. And this process we call arteriogenesis. Now, to give you an example of how this looks like, so the old rabbits already know the background, but for the newcomers or who haven't heard about that story, I just want to explain it in a few words. What you can see is a human heart with a completely occluded vessel. So everyone would expect that this patient suffered from a myocardial infarction. However, he did not. He did not recognize that this vessel was completely occluded, he had no problem with the heart, and even the physicians didn't know. So this was just found by chance. So what happened was that the stenosing process of this coronary artery developed very, very slowly. So the heart, the blood, which normally flows through this coronary artery, was redirected in one of the pre-existing collateral arteries. And when the process of stenosis increased, more and more blood was redirected into one of those pre-existing collateral arteries. And finally, this natural bypass occurred. So what happened? How does it work? What is the mechanism behind it? When the blood flow is redirected into those pre-existing collateral arteries, they experience increased mechanical load and increased shear stress. And this activates the endothelium of this collateral arteries, which finally results in the increased bioavailability of chemoattractants like monocyte chemoattractant 1 or TNF-alpha. So a lot of leukocytes are attracted, but most importantly, monocytes which mature to macrophages, and this macrophages allow the growth factors and cytokines the cells of the vasculature need to proliferate. So the whole process of collateral artery growth relies on an inflammatory process. And to show you in more detail how much this is dependent on innate immunity, here we're going to detail. So as I already mentioned, upon increased fluid shear stress, the endothelium gets activated. But what happened first of all? Due to the increased fluid shear stress, RNA is released from the endothelium. This is ribosomal RNA, which we find at the abdominal side of endothelial cells. And this extracellular RNA is now very important for arteriogenesis because it mediates the interaction of VEGF, vascular endothelial growth factor, with its receptor VEGF receptor 2 and neuropilin-1. So upon activation of this receptor, the von Willebrand factor is released. Von Willebrand factor now binds to platelet GP1B alpha and activates platelets. Now the activated platelets interact with neutrophils and create platelet neutrophil aggregates. Now neutrophils are also activated, and then there is an exacerbation of those activated neutrophils. Those neutrophils now produce cross-reactive oxygen species, which now activate mast cells. And now activated mast cells increase the bioavailability of TNF-alpha and MCP1, resulting in, on the one hand, in a feedback mechanism, recruiting more neutrophils, but on the other hand, also recruiting monocytes, which then mature to macrophages and supply the growth factors relevant for vascular cell proliferation, and finally the growth of this vessel. So we have a positive outward reward. So the most important thing for triggering arteriogenesis is this extracellular RNA, this ribosomal RNA. And I was thinking, is there a protein, or maybe it's some sort of protein, which might regulate the activity or the availability of this ribosomal RNA? And I went into the literature. And there I found, and thereby, of course, regulating arteriogenesis by binding this ribosomal RNA. And there I found a protein which is called SIRT, cold-induced RNA binding. And it is described in the literature, so with a very small sentence, that in silico analysis showed that the SIRT can bind ribosomal RNA. So I said, okay, this might be our candidate, and I will more in detail. So just for first, give you some more information about the SIRT. SIRT has many functions. So in the cell, it somehow regulates transcription, so it works as a chaperone. However, it also binds to mRNA and thereby regulates the translation. However, on special stress reactions or mild hypothermia, this protein is also released. And it's described that this extracellular SIRT acts as stem and somehow modulates inflammation. Now, to investigate whether SIRT plays a role in arteriogenesis, we use our hybrid model of arteriogenesis, which I want to explain to you. So, the rabbit hindlimb, the left leg, the right leg. Here is the femoral artery, and here you can see very faint those three existing connecting arteries. Also here. Now, on the left leg, we do a sham operation, means we open the skin, excise the femoral artery and weight, and just do the suture around the femoral artery without making a knot. On the right side, we do the same. So also here we isolate the vein, the artery, and the nerve, put the suture around the femoral artery. However, now we make a knot and close the femoral artery. So now the blood is redirected into those pre-existing collateral arteries, and due to the increased shear stress, they grow. You can see here very typical is corkscrew formation, so they have increased in length, but also in diameter, so the whole caliber of the vessel is increased. And this is a natural bypass of a functional artery. So this is our model. Now, using this animal model, we look what's going on with SERP. And I organized some sort of mice from Japan, which was not so easy. And we used arteriogenesis and with laser perfusion measurements. So before ligation of the femoral artery, of course, the perfusion of the right and left leg is identical, so it's 100% of each side. However, upon ligation, it drops to about 10% in the right leg. However, with the time, the perfusion increases again due to collateral artery growth. However, when you look at this picture, you can see SERP deficient mice. So in those mice, Arteriogenesis somehow worked better, significantly better. So I said, okay, is this intracellular SERP, is this extracellular SERP, or which SERP might they look? And now we did a second study and administered an antibody which resulted in depletion of extracellular SERP. And indeed, we got the completely same result. Also, when we depleted extracellular SERP, collateral artery growth, or let's say perfusion recovery, was significantly improved compared to wild type. So, but this might be also due to simple vasodilation. So we always look at the vessel themselves. So we did some immunohistological analysis. And first of all, we looked for the luminal diameter of the vessel, and we found that in SERP knockout mice, the luminal diameter is much larger than in wild-type mice. And this mice showed also an increased number of proliferating vascular cells of beard and eukaryotic cells. So there is indeed more vascular cell proliferation. So this is the same experiment again when was depleting extracellular SERP and completely extracellular cells. So extracellular SERP seems to be important for vascular cell proliferation. Now, what about the mechanism? So, as I already mentioned in the beginning, how does the whole story work? We have this extracellular RNA, which via the VEGF receptor 2 activates the endothelium. Von Willebrand factor is released, which activates platelets. And then we have this platelet neutrophil aggregate formation. And now we look for platelet neutrophil aggregate formation in SERP deficient mice, however, also in wild type mice where SERP was depleted, extracellular SERP was depleted. And again, we found in SERP deficient mice as well as in SERP wild type mice where extracellular SERP was depleted and significantly increased number of the platelet neutrophil aggregate. Now, of course, we look what's going on with muscles. Do we also find increased muscle activation? And this is indeed profound. So, SERP depletion as well as efficiency of SERP did not result in increased number of muscles, however, in an increased number of activated muscles. And finally, we looked for monocytes, for macrophages. As I informed you, we need those macrophages to deliver those growth factors and in muscular cells to proliferate. And indeed, we found in SERP deficient mice as well as in mice with where extracellular SERP was depleted, an increased number of macrophages, which is named for CD68, as number for macrophages. The number of so-called M1-like macrophages, which are more inflammatory, was not increased. However, the number of the so-called M2-like macrophages, MRC1 positive macrophages, which are responsible for regeneration, was significantly increased. So we are doing well. Now, altogether, this result indicate that SERP and in particular extracellular SERP promotes a whole sequence of cascade from the beginning from extracellular RNA, finally resulting in vascular cell proliferation and an increase in the diameter, so improved collateral outflow. So to summarize my results, so what is this book? What did we find? Cell deficient in mice, as well as the depletion of extracellular cell, resulted in an increased platelet neutrophil aggregate formation. Both also resulted in increased muscle activation and an increased recruitment of macrophages, in particular M2 like regenerative macrophages. The whole story resulted in increased vascular cell proliferation and supposed promotion of natural microvascular. So from this data we conclude that the cell, both intracellular but mainly probably extracellular cell, interferes with the process of arteriogenesis, so with the process of natural bypass growth. And depletion of extracellular cell might present a positive new therapeutic option to treat patients with vascular disease by promoting natural bypass. And I want to go.
Thank you very much, Lisa, for this clear presentation on a new responsive regenerative mechanism related to atheriogenesis. Other questions or comments from the audience? Okay.
Thank you very much. That was very elegant. There is an entire industry devoted to doing the opposite. I'm talking about the cancer therapeutics, the anti-VEGF. Would you have a concern in translating this to therapeutic options that you might inadvertently enhance vascularity of cancer? Something that
So I would say if people or if a patient already has cancer, I would be cautious. But if people do not have cancer, so to promote the growth of collaterals should not be harmful.
Yes.
Because it will not promote the growth of collaterals.
Thank you. Can I firstly agree with my colleague Malcolm? It's a great talk. Thank you very much. The patients that present with an acute MI have a varied collateralization. So some will have a massive collateral, some will have not much. So the actual stimulus for collateral growth, where do you put that? Why would there be such a variation?
I mean that the variation in collateralization is of course the genetics. Yeah? As you already said, some of them have more pre-existing collateral, the other one have only little pre-existing collateral. So if you have a lot, of course, an advantage. So, but we are not working on inducing the pre-existing, but on the growth of pre-existing collateral. And I think one of the major is now this particular organ, which activates the lecture receptor. So if we know very well if we apply lecture, we can apply cuts. Will not help, will not not at all.
My understanding has always been that a patient with coronary artery disease, that's the stimulus for mitotic division of cells and growth. Is that correct?
No, I don't think it's a stimulus, no. So from my point of view, as I said, it's a mechanical stimulus. We know very well that training, when there is training, we can improve also activity sectors. So if we do sports, but not to running like hell to get a medal, but daily a little bit, and this also improves activity sectors. We know very well from other studies. And therefore, I think it's really the activation of the epithelium and this part. And it was also very interesting to see that this extracellular but it's localized, so it's the green one. The red one are epithelial cells, of the green one is extracellular RNA of the growing collateral, and here this is a resting collateral of the germ operating side of the same animal. So in this RNA is abdominal, so it seems really to be an active process that this RNA is released. No passive, no damage of a vessel, but it's active release.
Okay, Sean, thank you. That was excellent presentation. It's not my field, so this may be a very naive question, but my understanding is the process is underlying angiogenesis, which is a very small vessel, can be quite distinct from arteriogenesis. And I think the question was a very good one on cancer, which, you know, obviously metastatic disease, we're talking about very small vessels.
So this is our model. We all know the function of arteries is to transport the blood through the body. The function of capillaries is to supply all the oxygen locally to the cells. So, and here, when we ligate the femoral artery, there is no more blood transportation to the lower leg. So the lower leg gets this key. And there we find angiogenesis, we find browning of the blood. And in the upper leg, due to increased shear stress, we find this collateral arteriogenesis. And this is also why we use that model of the heart, because the heart both mechanisms overlap. And we go from the mechanism to the further we belong to angiogenesis or to arteriogenesis. And this is a nice model for the arteriogenesis. The upper leg is the lower leg. And we need to control both parts of the body. And we also find if arteriogenesis is very well, we have less skin cells, and of course have less sensitive, because we need the arteriogenesis to be good. And if it's issue that
Okay, thank you very much, Lisa. We need to stop here with the discussion.
Question, Chairman.
Let me announce first a little housekeeping message. I got a sign from the organizers that we postpone the high tea after the next two talks, because otherwise it would be such a big interruption, etc. I hope everyone is
all agree.
okay. And I didn't tell the organizers, but it might be that there is also a side to the IT a bottle of rum, Mauritius rum. But so, Charlie,
I don't ask a question anymore.
Okay, Lisa, thank you very much.
Yes, ma'am, very nicely explained. I would just point out to another feature is that though you are promoting angiogenesis, but simultaneously you are activating a number of pro-inflammatory pathways. So that mediates atherogenesis, which further obstructs the lumen. So how would you explain that?
No, actually I didn't get your question very well.
See, you are increasing VEGF to increase the angiogenesis. That's increasing the circulation, the blood flow. But simultaneously, there's mast cell activation, macrophages are getting activated. A number of pro-inflammatory mediators are there, which can further accelerate atherogenesis and its complications. So that will further worsen the situation. These are two contradicting things happening. VEGF is increased area, and it has an immunosuppressive action. So it will suppress the immune reaction.
Ladies, I really hate to interrupt, but we also think about, you know, the schedule here. And I think suggested would be that you three sit together at the high tea, plus or minus one, whatever, and discuss the issue. Is that okay?
But maybe just this last word.
Lisa, please.
Last word is last sentence. If you think the macrophages have this M1, which are pro-inflammatory, and the M2, which are great regenerative. So it's not only bad, something they're all good.
Okay, so give her.
Thank you, Dr. Denai. What I didn't mention was that Dr. Denai is the leading expert in the field of atheriogenesis, and she is presently head of the atheriogenesis group at the Walter Brandley Center of Experimental Medicine and the Biomedical Center both LMU, Marche, Germany. Please give her another round of applause.
As mentioned before, Haiti has been shifted. I think it's on a wrong time zone. So we'll be moving swiftly on to the next presenter. So it's Dr. Ursad Yet-7. She earned her medical degree from Justus-Liebig-University, Faculty of Medicine in 2007. Afterwards, she completed a cardio residency in Istanbul University of Serapatha, Faculty of Medicine in 2013. In 2015, Dr. Yet-7 joined the cardio department in Koshek University. Alongside her clinical work, she pursued research studies, obtaining an MSL from the USC and a PhD in molecular medicine from Istanbul University. She primarily serves as a researcher in Koshek University with a main research focus on atherosclerosis. We would like to warmly invite her to stage to present her presentation on atherosclerosis effects of PCSK9.
First, I want to thank the organizing committee for inviting me this meeting and giving me opportunity to present my topic. And also, I want to thank the SSR Medical School for hosting us. And I want to talk about the atherosclerotic effects of PCSK9. As we know, the cardiovascular diseases are the leading cause of the death around the world. And atherosclerosis is the underlying pathology of cardiovascular diseases. Atherosclerosis begins with the endothelial dysfunction and accumulation of the LDL molecules under the endothelial barrier, and this triggers the inflammatory response and recruitment of inflammatory cells, monocytes, formation of foam cells, migration and proliferation of the vascular smooth muscle cells. All these processes causes atherosclerotic plaque formation. And atherosclerotic plaque can thicken the vascular wall, can cause luminal stenosis, or if it's structured, it can cause thrombosis. And in 2003, Abifedal and her friends discovered a molecule, PCSK9 molecule, and gain of mutation in the gene coding PCSK9 causes familial hyperlipidemia. And since then, the PCSK9 has garnered significant attention. PCSK9 is a family member of protein convertase family, and this family responsible for post-translational modifications of the proteins, and they are cleaving the peptide bonds. And PCSK9 is a member of this family, but except its autocatalytic activity, PCSK9 has no cleaving activity in the body as far as we know. And PCSK9 has prodomain and catalytic domain, and prodomain, after translation, prodomain cleaved and bound to the catalytic domain and inhibits the catalytic domain cleaving activity. And after that, PCSK9 has no catalytic activity. But the catalytic side of the PCSK9 can bind to LDL receptor, a cellular growth factor like A domain. And in the absence of PCSK9, LDL molecule can bind to LDL receptor, and they both goes into the liver cell in the endosomes, and in the acidic environment of endosomes, LDL goes to the lysosomes for degradation, but the LDL receptor turns and recycle to the cell surface. But when the PCSK9 binds to the LDL receptor, and the LDL and LDL receptor cannot be separated, they both go to the lysosomes for degradation, and LDL receptor recycling is blocked. This causes decrease in LDL receptors and increase in LDL levels. So it has been shown that LDL level and PCSK9 level are correlated, and both are increased in the familial hyperlipidemia patients. And since PCSK9 causes hyperlipidemia, various targets and various ways has explored to inhibit PCSK9. Small interfering RNA inhibits transcription of PCSK9, and small molecular inhibitors disrupt the processing of mature PCSK9, and monoclonal antibodies bind extracellular PCSK9, and adnexis also binds extracellular PCSK9. And we know that PCSK9 inhibitors now decrease the cardiovascular and cerebrovascular events. And in Fourier trial, it has been shown that the primary endpoint and secondary endpoints are decreased with the PCSK9 inhibitor monoclonal. And today, in daily clinical practice, we use PCSK9 inhibitors, monoclonal antibodies, and siRNA. So in GLOCAL trial, it has been demonstrated that the change in the total arterial volume was increased. The arterial volume decreased with evolocumab when added to the statin therapy. But it has been discovered that recently it's recognized that the PCSK9 level is correlated with the atherosclerosis, not only with its hyperlipidemic effect, its atherosclerotic effects totally independent from its hyperlipidemic effect. And in Atheroma iris study, it has discovered that the negative core fraction of atheroma plaque was increased with the PCSK9 level regardless of LDL levels. And also in a cohort study, it was discovered that PCSK, when LDL levels were adjusted, the increment in PCSK9 causes total plaque area increment. And this trial has highlighted that PCSK9 has direct atherosclerotic effects on vascular wall cells, and PCSK9 has effects on macrophages, endothelial cells, vascular smooth muscle cells, and also these cells can express PCSK9. And monocytes enter the endothelial, under the endothelial barrier, and its uptake modified lipids and turn into the foam cells, and scavenger receptors mediate this uptake of the lipids, and also ABCA1 receptor transporter causes efflux of the cholesterol out of the macrophage to protect macrophage to forming foam cells. And PCSK9 induces the scavenger receptor expression, and PCSK9 inhibits cholesterol efflux from macrophages by reducing ABCA1 protein expression, and thus it contributes to formation of foam cells. And also, scavenger receptors are not only the responsible for lipid uptake, they are also inflammatory. They have also inflammatory actions. They cause increase in the inflammatory cytokines, and they activate NF-kappa B. And LOX1 is the best known inflammatory scavenger receptor. And PCSK9 induces LOX1 expression also in endothelial cells and smooth muscle cells. And when oxidized LDL induced the PCSK9 LOX1 expression, silencing of PCSK9 with its siRNA causes decrease in LOX1 and NF-kappa B expression. And also silencing LOX1 causes decrease in the PCSK9 expression. And NF-kappa B is an important transcription factor, and it causes transcription of inflammatory cytokines. And PCSK9 also stimulates the activation of the and nuclear transportation of NF-kappa B. NF-kappa B normally stays in the nucleus with its inhibitor. When it's activated, it separates from its inhibitor and heterodimer enter the nucleus and causes inflammatory cytokine production. So PCSK9 stimulates nuclear transportation of NF-kappa B, and PCSK9 also induces the expression of inflammatory cytokines in the macrophage. And silencing PCSK9 reduces the expression levels of the inflammatory cytokines in atherosclerotic alphas of apoptosis. And also PCSK9 involved in the apoptosis process. BCL2 and BAX are two proteins that are take role in the apoptosis, and PCSK9 inhibits BCL2 and promotes BAX. And PCSK9 siRNA increases the expression of BCL2 and decreases the expression of BAX. And when we silence the PCSK9, also the caspase-9 and caspase-3 expression was decreased, and silencing of PCSK9 with siRNA inhibits apoptosis in endothelial cells. And also lipopolysaccharide increases PCSK9 expression in muscular cells, and this is most pronounced under low shear stress. Shear stress is the tangential stress that blood flow applies to the vascular wall, and low shear stress occurs in the branching of the arteries and the bifurcation regions, and these regions are more prone to atherosclerosis. And under the low shear stress, PCSK9 response to the lipopolysaccharide was most pronounced. And endothelial cell and vascular smooth muscle cell crosstalk also important in atherosclerosis. And when endothelial cells exposed to atherogenic stimuli, they can cause increase in PCSK9 and NF-kappa B expression in the vascular smooth muscle cells. And we applied oxidized LDL, hydrogen peroxide, and cigarette smoke extract to endothelial cells. And then we incubated with the supernaturals of these cells, the vascular smooth muscle cells, and in vascular smooth muscle cells, we observed PCSK9 expression increments and also NF-kappa B expression increment. And up to this point, this data that I shared was all well known and agreed, but there are also some conflicting data on vascular effects of PCSK9. First of all, Ferry and his friends published a paper about the PCSK9 knockout mice. They applied perivascular parotid collar placement to these mice. This is a model to study intimal hyperplasia and creating a stenosis in the carotid and evaluating the intimal hyperplasia. And they saw that in PCSK9 knockout mice, near intimal hyperplasia was much more lower than the PCSK9 positive mice. And in mice with allograft aortic transplantation, PCSK9 knockout significantly reduced vascular stenosis, intimal hyperplasia, and collagen deposition. However, a very recently published paper, they implanted stent to the mice, and they observed increase in intimal hyperplasia with alirocumab. Alirocumab is a monoclonal antibody to PCSK9, and they observed increased stenosis with alirocumab. And also they evaluated wound healing assay, and they saw with alirocumab decrease in the vascular, decrease in the proliferation and migration in endothelial cells, and increase in proliferation and migration in vascular smooth muscle cells. So these conflicting data can be due to different PCSK9 inhibition mechanisms, because the studies that show the increase in the intimal hyperplasia, this study, it was extracellular PCSK9 inhibition, but the studies that show that PCSK9 inhibition causes decrease in the intimal hyperplasia, they use PCSK9 knockout models, that is intracellular PCSK9 inhibition. And the data that approved that was published in ischemia reperfusion model. PCSK9 knockout mice were protected from ischemia reperfusion injury. However, with PCSK9 inhibitor, their protection couldn't be demonstrated, and the impact size was decreased with PCSK9 knockout, but didn't decrease with the extracellular PCSK9 inhibitor. And also there's mixed evidence regarding the PCSK9 effect on macrophage activation is dependent on LDL receptor. In LDL receptor negative macrophages, it has shown that PCSK9 couldn't cause the increment in the TNF alpha level. However, in another study, the PCSK9 the LDL receptor knockout mice, PCSK9 causes increased inflammatory cytokines. So PCSK9 plays role in the degradation of LDL receptor family, except LDL receptor, site LDL receptor, LRP1, LDL receptor, VLDL receptor, and E2 receptor. And it was suggested that in vascular smooth muscle cells and endothelial cells, PCSK9 interacts with the LDL receptor and LRP1 receptor, but these interactions, the consequences of these interactions is not very well known. So we conducted experiments about the dependence of the atherosclerotic effect of PCSK9 on vascular wall cells, whether it's dependent on VLDL receptor or LRP1 receptor. We conducted immunofluorescence experiments. First, we labeled the NF-kappa B antibody with green, and blues are DAPI for nucleus. And in control, the NF-kappa B antibodies were in cytoplasm, and the nucleuses were empty. And we used positive control, TNF-alpha 20 nanogram per milliliter. And after 2 hours of incubation, the nuclear translocation observed with TNF-alpha. And also, we incubated cells with 2.5 microgram milliliter PCSK9. And after 2 hours, we observed again the nuclear translocation of NF-kappa B. And then we applied siRNA to the cells, LRP1 and VLDL receptor siRNA. And we demonstrated the decrease in the gene expression with qPCR and decrease in the protein expression with Western blot. And as expected, and we applied PCSK9 again to the cells. And as expected, with control siRNA, the nuclear translocation happens again. And with LRP siRNA, the nuclear translocation is observed also. But when we applied VLDL receptor siRNA, with PCSK9, the nuclear translocation in NF-kappa B was inhibited, and the nucleuses was empty, as you see. And when we silent VLDL receptor with LRP receptor together, then the with PCSK9 nuclear translocation of NF-kappa B was observed again. And this can be caused by the LRP1 is an anti-inflammatory receptor. It inhibits normally the NF-kappa deactivation. So adding LRP1 silencing to VLDL receptor silencing blunted the protection effect of VLDL receptor silencing. So if I summarize, we know that PCSK9 induce inflammatory state and also apoptosis in endothelial cells. In macrophages, it induces inflammatory cytokines. And until today, we were believing that in vascular smooth muscle cells, it promotes proliferation and migration, but now it's in debate. And also in vascular smooth muscle cells and endothelial cells, we know that it interacts with the VLDL receptor and project started to clarify PCSK9's effects on vascular wall cell proliferation, the difference between extracellular PCSK9 inhibition and inhibition of PCSK9 expression, the receptors of its direct atherosclerotic effects, and further mechanisms to its direct atherosclerotic effects. And I hope I will present some of the data in Istanbul in 2024. Thank you for your patience.
Thank you very much. Actually, we have 2 minutes for a few questions. Okay, Mike Humphrey.
Thank you very much. That was very comprehensive, and I'm not sure that I comprehended all, but thank you. One of the issues that perhaps you can correct me if I'm wrong, if you're suggesting that foam cells are more likely to accumulate, are you implying that whatever atheroma is there will become less stable or more likely to, because that has very important implications?
With the foam cell formation, the atheroma plaque become more unstable, and so many studies have conducted with PCSK9 or non-PCSK9 to inhibit the foam cell formation. So yes, and inhibiting PCSK9 reduces the foam cell formation. Yes.
Yeah. Please.
Hi, very nice talk and also a very interesting proposal. I have one question regarding your expression or extracellular inhibition, because the siRNA data, those siRNA used clinically, it's GalNAc conjugated siRNA. Actually, it's primarily targets the hepatocytes instead of circulating the systemic PCSK9. But the antibodies could target, the circulating or, systemic PCSK9. Do you think that's the difference outcome reported is because of the cell type they are targeting instead of expression or extracellular PCSK9?
Yes, it can be. In these experiments, the PCSK9 knockout mice were used instead of siRNA, So you are right in siRNA that used in clinical is tend to go to hepatocytes and the extras, but still, if we silence the PCSK9 expression in the vascular wall cells, maybe the effects will be more protective. And extracellular yes, we are inhibiting the extracellular PCSK9, but extracellular PCSK9 and intracellular vascular expressed PCSK9 has maybe don't have the same effects.
Thank you.
Okay, thank you very much for this interesting session. We are approaching the final talk of the first day by Belle Rive, and the young organization team will introduce.
Now we'd like to have Professor Immerman MD, Free State, Schareit, Berlin, Germany, PhD, Cell Physiology at Ludwig Maximilian Universität, LMU, Mainz, Germany, postdoctoral work in lipid biochemistry at the University of Utrecht, the Netherlands, Assistant Professor, Institute of Physiology, LMU, and his major research topic is immunothrombosis. Thrombosis has immune mechanisms in infection and metastasis. Today, he will be discussing with us the lymphocyte is to neutrophil ratio as predictor of thrombosis. Please, a round of applause for Professor Engelman.
Yes, I would like to thank the organizers for this very kind invitation, and I'm very happy to be here in Mauritius, which I find not only a very beautiful country, seems to be a very nice example of a society, so at least at my first view, that aims to integrate diverse cultures and diverse religions. And I think nowadays this is especially important, having the situation that we have currently. Okay, so I'm talking about CD4 positive T cells and their role for thrombosis in infection. Now, where are the pictures? That's very strange because they were there, then there's nothing, because we checked them together beforehand and everything was there. I think I can get it on the desktop. That's true. That's the problem. I have another one. Now it's there. Okay, so that looks far better. I mean, recently, especially in Europe, it's much clearer that thrombosis is a very important aspect of systemic infection, especially. And here I have given two examples of systemic infections that give rise to thrombosis. One is very well known, sepsis as induced by, for example, gram-negative bacteria. And then that's how do you want to call it, a special form of infection, SARS-CoV-2 infection. And interestingly, what we proposed a couple of, actually one ago, has been quite nicely demonstrated to occur under this condition, especially SARS-CoV-2 infection. So what are the essential elements of this concept of immunothrombosis that we believe is a physiological mechanism of thrombosis? It's very important to keep this in mind. We talk about physiological mechanism and we talk about a pathological aberration, which is happening in this type of infection and also infection. And two major elements here are innate immune cells, namely monocytes, now are classical monocytes. And these classical monocytes expose tissue factor. Tissue factor is the main starter protein of blood circulation inside the blood vessel. So we have this protein was called tissue factor because it was thought to be expressed mainly in the subendothelial space when it came out in about 20 years ago that it is also expressed by immune cells inside the vessel. And so this is an important element. Here the classical monocyte, which expresses tissue factor, and then there's a second major innate immune cell, this is the neutrophil. The first to show in 2010 that the neutrophils release nets to increase thrombosis by various mechanisms. I don't want to go into detail here. One of the mechanisms is degradation of the natural antioxidant pathway. There are other mechanisms, for example, activation of the contact pathway. Now, the question is, if this is a physiological process, how is this process being restricted? We all know that when thrombosis occurs, there's always a threat of vessel occlusion. I've heard a lot about vessel occlusion here, and so it's very important to know which mechanisms are available to restrict thrombosis. And one of the mechanisms was mentioned by Sean Jackson before. Even when you think about mechanisms that inhibit thrombosis, because the endothelium expresses anticoagulants and which activates protein C and it activates fibrinolysis. So, and what has also been recently, and it has also been a current theme in this session, activation of the complement. There's a lot of interaction going between complement and coagulation pathways. So the main question we were asking was, are there any immune cells, not endothelium, are there any immune cells that express microvascular thrombosis in this practice? Because this is much less investigated than the prothrombotic function. The immune cells plus the monocytes in the tract. Now, so we did a rather comprehensive study to look at the immune cells that are being recruited during systemic infection. We have here a model of systemic infection which doesn't lead to grave organ damage. So it's a physiological form. And as you can see here, it's not unexpected to see neutrophils being recruited, to see classical monocytes being recruited, non-classical monocytes as well. And then there are CD4 positive T cells being recruited at almost the same time window as the innate immune cell. Surprised us a lot. Because usually, if you think about adaptive immune cells, they come later. Because usually you think there's a first period of innate immunity, and then there's training of the immune system by adaptive immune cells. That takes much longer. And then you have to characterize this subtypes. We found that most of them were Ro-gamma positive, meaning they were Th7 T cells, but they were also Th2 cells, Tregs, and a combination of. So we asked ourselves, what can we see positive during and we did a very simple, unbiased screen of these T cells in early infections, transcriptomic screen, and what came out was first was innate immune response to the network, the second was blood coagulation fibrinolysis. So that was also quite exciting. Because if you think about the adaptive immune system, you think that this is doing innate immune responses. But it is clear that in the last 20 years, many studies have shown that also CD4 T cells can have inflammatory effects. And among the blood coagulations, when we looked at the genes that were mostly involved, and these were negative regulators of coagulation, as for example, plasminogen. So it appears that there's a messenger RNA for plasminogen, quite highly expressed in the others. So we went on to look at whether this transcriptomic data was important for indicated for changes. And this summarizes what we found. I mean, here we have looked at tissue effect I mentioned earlier, this pretty fast blood circulation. And we found that, which is well known, that the cells that are positive for tissue effect are either classic monocytes or neutrophils. Whereas the other cells, especially the C-positive cells, not unexpected. However, when we look at, for example, cardiovascular carcinoma, we found that the C-positive the highest expression of fibrinolysis regulator. This didn't hold only for plasminogen, but also for many other fibrinolysis, for the major fibrinolysis regulators. UPA, TPA, here's an example for UPA positive again result for the subtypes, and again we see that H7 are the most important ones that express fibrinolysis. So the next thing what we did was we asked ourselves, where do these TPH17 cells in the liver come from? And here our cooperation partners used the so-called house model to categorize all cells that are renal green. And then you have the possibility by nuclei activate specific cells and specific organ. Since TH17 cells are most present in the small intestine, to activate cells, so we specifically labeled the TH17 cells in the small intestine and then looked at the time point when these cells will arrive in the liver. And we saw indeed that iron 78 positive so meaning TH17 cells were in increasing amounts recruited to the liver, but not to the lung. Okay, so this was a liver specific finding. We all know that the small intestine is linked by diverse vessels to the liver. Next question was whether this this impact. Okay, here's an image. Okay, whether the presence of the fibrinolysis regulators to the surface of the C-positive T cells was relevant for fibrosis for development of thrombosis. And here we found that fibrosis deposition in the microcirculation in the liver was strongly increased, and this was also apparent for the number of microthrombi. So this confirmed that somehow and we did this also in another model of systemic impact, not using E. coli, but the ground positive, another agent of sepsis, pneumonia. And here we saw also that depletion of positive T cells led to an increase in fibrosis. And this is just a picture to show you the antagonistic effect of the other cells versus neutrophils plus this is demise of neutrophils. Here we see that specifically of plastic. We see exactly the contribute of that. And this tells us that we think that the so-called leukocyte ratio, at least for which is also quite important for this prediction, is due most likely to this antithrombotic effect of the T cells versus the prothrombotic effect of the immune cell. Now we ask ourselves, what was the mechanism? How did these cancer cells decrease? And as we had seen, increased expression of negative regulators and the transcriptomic and protein analysis. So we looked at simple fibrinolytic assays, plasma information assays. Here we took the CD4 positive T cells from activated, from infected mice and non-infected mice. And here we used human T helper cells. And so we come to the helper cells via the TCR pathway, via the T cell receptor. We see a very strong increase in fibrinolytic activity on T cell activation. We see both the human and the mouse. So to further analyze this, we looked at we isolated the answers from mice, activated them in ex vivo, and treated them with very known, very old hydrolysis inhibitor acid. And as you can see here, when we block this, the inhibitory effect of the other cells is completely blocked. So it appears that indeed the other cells mediate the anti-thrombotic effect via stimulation of fibrinolysis. And these parts of the cell. So then the next step was, how does this occur and where does it? And to our surprise, we noticed that cell activation preferentially occurs in microvascular. Then we have labeled fibrin before and interleukin 2, which is the indicator of cell activation. And as you can see, these four they only develop practically only develop fibrin. So when T cells encounter fibrin, and to confirm this, we use factor 12 knockout. So why we lack the contact pathway in factor 12? We see that the recruitment and activation of the CD4 positive cells is reduced, whereas when we use plasminogen mice, where we could show that they have an increased fibrin, the activation and the arrest is increased. As another indicator, we use the factor 10A inhibitor rivaroxaban. Now we have a very successful anti-thrombotic therapy the last 15 years by so-called direct oral anticoagulant. Successful, and when we apply this to mice, we see that due to the fibrin reduction, we see a strong increase in activation of diseases. The same holds for mice. So fibrin deposition clearly regulates cell activation. What type of T cell activation is this? So we have two types of T cell activation. Very roughly spoken. One is the very well known antigen-independent or prime T cell activation by MHC class 2 presenting antigens. And another one which has come into focus in the last 10 to 15 years is something which is called cytokine activation of T cells or also bystander. And as you can see here, both blocking and the MHC2 antibody specifically interaction between the peptides and MHC2. And this antibody specifically blocks the cytokines that activate T cells. So both seem to be part for T cell activation in early infection. Remember that we are always talking about early infection in the first 3 to 6 hours after diffusion of bacteria. Start of bacterial infection. Okay. Then since we had shown that fibrin deposition activates T cells in people in mice, we want to look whether it also happens in humans. So we use human CD4 and integrate them on slides coated with either organism with fibrin, and see here that a strong increase in T cell activation when the T cells are coated on fibrin as compared to the control. And more importantly, this effect can be almost fully blocked by LFA1 inhibition. LFA1 is an integrin, very important for T cells. And we could show that this fibrin-dependent T cell activation is almost completely blocked by a neutralizing antibody against LFA1. At least in the human system. And this holds also for the mouse system. With this complicated slide here, let's start here. This tells us that blocking LFA1 also reduces fibrin-dependent T cell activation in vivo, in mice and people. And here we have analyzed by ultraviolet imaging of opposite T cells in the microcirculation, and we have found that both activation and migration of T cells is dependent on them. I will skip this slide, I guess. Yeah, and just to close here my talk. The application to different bioinfection, we have compared SARS-CoV-2 infection with influenza virus. So we have analyzed of from 12 patients with 2 and 9 patients with influenza virus. Here, in the SARS-CoV-2 patients, we have clearly much lower the CD4 in the microcirculation, which we could call lymphopenia as compared to influenza virus. And at the same time, however, the number of microthrombi strongly increased in COVID-19 as compared to influenza virus. So, meaning, together with the experimental data in mice and the human data, that the lower the CD4 in the microcirculation, the higher the number of thrombi. So there is a clear negative correlation between the presence of CD4 positive cells and thrombosis. Okay, so this summarizes my talk. Fibrin attracts the other cell, mediates the LFA1 dependent activation in a systemic infection. Activated CD4 positive T cells suppress fibrin, thus mediating negative feedback mechanism on microvascular thrombosis. So the other cells are mediators of a negative feedback. They directly and this could indicate that leukemia might facilitate thrombosis during human infection. And with this, I would like to thank the different fellows for the study, and Immuno-thrombosis and other cooperation partners, especially also Klaus is involved, and from Munich. Thank you for your attention.
Thank you, Belle, for this new story. Whenever we see on a meeting there is another cell, another mechanism that contributes to immuno-thrombosis, your favorite subject in the last years. Thank you very much. So we have a little bit time for some questions or comments in the audience. So, thank you for an elegant presentation. First question, the interaction between the T cells with the fibrin. This is an LFA1 binding the fibrin, you think? Is correct, because alpha-1 obviously binds fibrinity.
So we don't know. We have in the immune system, we have just the T cells and the fibrin. And this can be blocked, this activation can be blocked by blood. So in this system, sure, that is a direct interaction between the integrin and fibrin. But in the mouse system, it's obviously much more complicated to demonstrate.
My second question, obviously fibrin binds a lot of thrombin. And is there a role for the thrombin when you block power receptors, for example, on the lymph cell?
So far, we have blocked part 1 and part 2. We don't see an effect. And what I haven't, because I couldn't get into much detail, but certainly played a big role also. So it's not just the fibrin in vivo, but in vivo players are also massive.
Yeah.
Thank you, sir. It was actually I just curious about this, whether it can be stopped by using steroids. It causes thrombosis. So can the process be stopped, blocked?
Okay. Meaning if you inhibit inflammation with steroids, is this what you mean? If you inhibit inflammation with steroids, that would, because of the pro-thrombotic function of the inflammatory cells, that is easily understood to decrease thrombosis.
So can it be a preventive for thrombosis? Steroid.
Yes.
Right. Thank you.
Okay. Other questions?
So thank you very much, all the speakers and discussants. I think it was a pretty interesting session. And now the high tea session is open. Okay, thank you.
First, I'd like to call Professor Klaus Tresner to present the following certificates to our wonderful speakers. First, we'll call upon Professor Elizabeth Denai. Professor Engelman.
Can you please call on stage Professor Sean Jackson? And doctors.
Now, I would like to call upon Professor Derek Hunsley to provide Professor Tresner with the certificate. Now, with that being our final speaker, this concludes the focus of today's discussion. I would like to thank you all for your time and attention here today. We will resume back here tomorrow at 9:15, and please scan our QR code and give us some feedback on our function here today. We wish you safe travels. Have a pleasant evening, and I look forward to seeing you all here tomorrow. Now let us break for tea. Thank you.